So we're going to talk about the urinary system, first focusing on the anatomy, which is pretty extensive with the kidney and is very important to how the kidney is able to carry out the functions that it does. Okay, so talking about the organs of the urinary system, the one that we talk about the most, that we focus on the most, are the kidneys, which of course we have two of them, a right and a left. The kidneys are what filter blood and remove things that could potentially become harmful, like metabolic waste products. They also can decrease or increase the amount of water or other solutes that we excrete or reabsorb. So they are part of helping us to maintain fluid in our body, water, and electrolytes such as sodium and potassium and chloride. It also helps with the acid base by allowing us to excrete hydrogen ions or bicarbonate, and helps us to maintain blood pressure, which we've actually talked about through the Ross system. Remember the renin, angiotensin, aldosterone system. So the urinary tract is basically the organs which carry urine to exit the body. So carrying urine from the kidneys, you have the ureters. Ureters carry urine to the bladder, which will store it, and then the urethra takes urine to its pathway out of the body. Ureters are on the posterior portion of the kidneys. They exit right where you have the indention, the hilus or hilum, and they empty into the posterior surface of the bladder on the floor. The urethra exits at the very bottom of. So urine will be formed in the renal pelvis. It will move down the ureters, and the dotted line is to show that these are going behind the kidney, and they open up so it can fill the bladder, and you have a right and left ureter. So as the urinary bladder is going to get full, then the sphincter, which we'll talk about this later, opens, that allows the urethra and for urine to exit the body. Bladder. So again, the kidneys are primarily what we focus on because that's what carries out most of the functions of the urinary system, those that involve keeping our body in homeostasis. So again, it filters the blood, so it helps remove metabolic waste. We'll talk about these more in depth, but these are acids and mainly nitrogenous waste. It also regulates fluid and electrolytes by controlling the osmolarity of our blood, looking at how much fluid per electrolyte, and then acid-base balance of the body, with the same buffers that we talked about with the digestive system. So carbonic acid can be split, and we can decide to keep or secrete hydrogen ions, keep or secrete bicarbonate, depending on what the current pH is. Kidneys influence blood pressure directly by secreting an enzyme, which is going to be renin, which affects blood volume. And remember, renin activates angiotensin, so that will help increase peripheral resistance, which increases blood pressure as well. It also regulates blood pressure by controlling bone marrow production of red blood cells. Obviously, the more cells, the more viscous the blood, the higher the blood pressure. And this, of course, is done through erythropoietin. Other important functions include detoxifying substances in the blood. Again, primarily these are nitrogenous waste. Of course, some of the waste from damaged red blood cells occurs as well. Activate vitamin D into calcitriol, and also making new glucose, which is gluconeogenesis. So we will discuss some of these in more detail later. So the kidneys are positioned very posterior dorsal portion of the body. They are, a small portion of them is protected by the ribs. Not much. course, you can see how the ureters again dump into the posterior portion of the bladder here. Also, you can see how the right kidney is slightly lower than the left. And that's because, of course, the placement of the liver. Your lab book actually shows the position of this. It makes it a little bit more obvious in that picture. It's kind of hard to see in this picture. So review, the urinary system consists of the kidneys, which of course filter the blood. The ureters will take urine from the kidneys to the bladder. The bladder stores it and then ejects it into the urethra where it allows it to exit the body. So of course those are all organs of the urinary system. The ureters, bladder, and urethra would constitute the urinary tract. Which of the following is not a function of the kidneys? They do get rid of any ions that we have in excess. That's how we control electrolytes. They do release erythropoietin when red blood cell count is low, oxygen is low, or when blood pressure is low, and they do activate vitamin D. Bile production, of course, is done by the liver. Because of the position of the liver, the left kidney is slightly lower than the right. Of course, that is false. It's opposite. The right is slightly lower than the left because of the liver. Okay, now we're going to focus on the actual anatomy of the kidneys themselves. So the kidneys have a lot of extra protection, including 3 layers of connective tissue. So these are listed from superficial to deep. So the most external is the renal fascia. This is dense, irregular connective tissue. It holds the kidney to the back posterior portion of the peritoneum and also to the most posterior portion of the abdominal wall. The capsule fat adipose tissue is the thickest layer, and it basically cushions, surrounds the kidneys. Fat does a lot of protection for hard blows, protects from physical shock. And then the renal capsule, which is closest to the kidney, it's dense, irregular connective tissue, and it protects it from infection and physical trauma. So here it's a transverse cut, and then you're looking from the bottom, an inferior view. So this first layer, which was your renal fascia, it's the very outermost portion. Again, it helps anchor it to the back of the wall, as we see here, and the back part of the peritoneum. All the yellow that you see there, which is of course the thickest, that's the adipose tissue, which provides a lot of protection, as well as you can see there's a lot of muscle around the kidneys as well. And then directly adhering to each kidney would be that most internal layer, which would be the renal capsule. Kind of like a lima bean peeling off. You could see that on the dissected kidneys. So the kidneys are about a bar of soap once you remove all of those 3 layers of connective tissue that surround it. The indention is called a hilum or hilus. This is where the renal artery enters, where the renal vein exits, where nerves enter that serve the kidney as well, and of course where the ureter exits. The hilum opens up into a space called the renal sinus. And this is an area where we collect urine and basically start to funnel it towards the ureter, and it has a lot of adipose tissue in it. Okay, 3 distinct regions can be seen. The outermost cortex, the middle is the medulla, and the very innermost are the pelvis. So the cortex and medulla are actually where the functions occur. This is where we're forming urine. Basically, that's where the nephron's going to be. Where the pelvis simply helps drain urine and funnel it into the ureter. Okay, so the renal cortex is a dark color if you see a fresh one from a specimen because of all the blood vessels. So 90 to 95% of all the kidney's blood vessels are actually within the cortex. For the rest of the anatomy, I'm going to use the picture. Okay, so again, the very outermost here, this would be the renal capsule. The adipose layer and the renal fascia have already been removed. Okay, so between the two black lines is the cortex. And of course, I didn't continue that black line all the way around, but that very outer ring is the cortex. Again, this is where most of the blood vessels are found. Then between that black line and the red, that constitutes the medulla, which you have two primary structures in the medulla. The triangular or pyramidal shaped structures are called renal or medullary pyramids. And then the space between the pyramids, which we refer to as a renal column, which, as you see in this picture, it's similar to the tissue of the cortex. You're going to have a lot of blood vessels going through here, the interlobar vessels. So again, these structures are where we are making urine. So we're filtering the blood, basically deciding what we want to keep in the body and what we want to get rid of it. Underneath each of the renal pyramids, we have a small funnel-shaped structure called a minor calyx. So this will collect all of the urine that's made by the nephrons within this renal pyramid. And where several minor calyces come together, that is what we call a major calyce. So they're very, very small. They're basically just right before you have that renal sinus, which is the space in which the urine filters. The pelvis is the area. And of course, urine will form here, and then it will be taken out of the ureter. And of course, what is forming the urine are these highly microscopic structures, which we call a nephron. And you have about 1 million per kidney. Okay, blood supply to the kidneys is very complex. The right and left renal arteries come immediately off of the abdominal aorta. And of course, the right and left renal veins will dump back into the inferior vena cava. Kidneys receive about 1/4 of the total cardiac output, or about 1,200 milliliters of blood per minute. So that's how much blood they are filtering through. Renal arteries will basically branch into smaller and smaller vessels as they make their way to the renal cortex. So these are the vessels in the order, basically as you enter the hilum until you get into the cortex, which I'm going to use the picture to talk about these. Okay, the kidney is unusual in that the capillary bed is fed by an arteriole, which means blood enters it through an arteriole, and it's also drained from an arteriole. And that's what actually allows the glomerulus, which are the capillary beds of the kidneys, to withstand a greater pressure, is that they have an arteriole draining it. So the glomerulus and the capillaries are held within a renal capsule. So the glomerulus and the capsule that surrounds it comprise the renal corpuscle. Efferent arterioles will then deliver blood into a second capillary bed. Typically, it stays within the cortex. You're talking about the peritubular capillaries, although sometimes it does go into the vasa recta. Okay, so to talk about the blood vessels, if we use the picture, just like when you're learning the names of arteries and veins, every time you have a branching, you're going to change names. So the renal artery is coming directly off of the abdominal aorta. So as soon as that branches, kind of see 3 little ones here, that is the segmental artery. The segmental artery branches into the interlobar. To be clear, interlobar extend all the way through the renal columns. Arching over each medullary pyramid, kind of the border between your cortex and the medulla, is your arcuate artery. Extending from each arcuate artery would be a cortical radiate artery or an interlobar. So in this picture, we're zooming in on where the medulla and the cortex come together. Okay, so blood supply to and from the kidneys. So the abdominal aorta would be about here. So extending from the abdominal aorta, this would be the left kidney. So this would be your left renal artery. And just like when we were naming blood vessels initially, every time you have a branch, you're going to change the name. So as soon as the renal artery branches, we now call these branches the segmental artery. Segmental arteries will branch sometimes within the upper portions of the pelvis, but they do extend all the way through the renal column. So any arteries within the renal columns would be your interlobar. Interlobar will then arch over kind of this boundary between the medulla and the cortex. This is what we call the arcuate artery. Coming off of the arcuate artery, you're going to have several thousand cortical radiate or interlobular arteries. And if we look on the blown up picture here, the arcuate artery, coming off of that would be the interlobular or cortical radiate. Now you have the first arteriole that's going to feed into the first capillary bed of the kidney. So this represents the afferent arteriole that feeds directly into the glomerulus. Efferent arteriole drains it, takes blood into typically peritubular capillaries. Peritubular capillaries will drain back into your interlobular vein or cortical radiate vein, which will then dump into the arcuate. So cortical radiate, interlobular, arcuate vein, interlobar, and you'll notice that the interlobar don't branch again until the renal vein. So you do not have segmental arteries like you did. You don't have segmental veins like you did with arteries. You need to know the blood flow. So the nephron is the functional unit of the kidney. Again, you have about a little over 1 million of nephrons in each kidney. So two main parts, the renal corpuscle and then the tubule. The corpuscle is where we filter blood, and the corpuscle is comprised of two parts. The glomerulus, which is actually the capillaries. If you remember, we were doing blood vessels, fenestrated, are more permeable than normal continuous capillaries, and that's the type that we have in the kidneys. And then what surrounds the glomerulus, which is the glomerular capsule or Bowman's capsule, which has an outer parietal and inner visceral layer. For the rest of the structure, okay, so the corpuscle is made up of Bowman's or the glomerular capsule, which consists of the parietal layer, which is made of simple squamous, and that surrounds the glomerulus. And then closely adhering to the glomerulus is the visceral layer. The visceral layer is actually made up of these yellow cells. So the layer is visceral, the cell is the podocyte. And podocytes have these little feet called pedicels. And the pedicels of one podocyte will interdigitate with the pedicels of another. And between those spaces is where fluid will leave the blood capillaries, which is the glomerulus, and be forced into the space between the visceral and the parietal layer. This is Bowman's or glomerular space, and as fluid accumulates here, we call that fluid filtrate. And so this filtrate will collect and it will enter the next part of the nephron, which includes the tubules, which is going to be the proximal convoluted tubule. So feeding the glomerulus, you have the afferent arteriole. So blood's going to come in at a high pressure. The high pressure in the glomerulus is what forces blood to leave, which is what we call filtration. Not all of the blood and fluid in the glomerulus will exit out through filtrate. So that remains will exit through the efferent arteriole and will then feed into the peritubular capillary beds. Okay, so the next part, so this whole portion again is the corpuscle. So that includes both parts of the capsule and the glomerulus. So after the corpuscle, you have the parts of the nephron. So the filtrate will enter what we call the proximal tubule or proximal convoluted tubule. Both are used. Then it will enter this elongated structure, the nephron loop or loop of Henle. So you have the descending limb of the loop, which carries fluid downward, the ascending, which fluid will move upward, into the distal tubule or distal convoluted tubule. The filtrate will then be dumped into the collecting system, which first part is going to be the collecting duct. So these other little branches here, these represent where other neurons would also be dumping the filtrate from their distal convoluted tubules. In the first part of the nephron, the proximal tubule or proximal convoluted tubule, it's the longest segment. It has some straight portions, but most of it is coiled or what we call convoluted. It has simple cuboidal cells that have microvilli on the surface. These create a brush border, and microvilli are going to increase the surface area, just as they increase the surface area in the small intestines. Why would we want to increase the surface area? To help with reabsorption, which is the main function that occurs in the proximal convoluted tubule. The nephron loop or loop of Henle is straight except for the bend as it starts to move back up. And it's the only portion of the nephron that leaves the cortex and extends within the medulla. So again, as fluid moves down, you have the descending limb and you have the ascending limb. The descending limb is also known as the thin limb because it is made of simple squamous epithelium. The ascending limb changes to simple cuboidal. And unlike the proximal convoluted tubule, it does not have microvilli. So the ascending is a lot thicker. The loop is still thin, but the ascending portion is thick. The distal tubule, also like the proximal tubule, has straight and coiled regions, and it will dump the filtrate directly into the next part, which is your collecting system. It has simple cuboidal epithelium with very few microvilli, not enough to create a brush border. Okay, if we look close to the renal corpuscle and look at the distal convoluted tubule, there is a structure which we see, which is called the juxtaglomerular apparatus or complex. And it is made up of cells that surround the afferent and efferent arteriole, as well as cells that are found within the distal convoluted loop. Macula densa constitute the cells that are very tightly packed together in the distal convoluted loop, and then you have special smooth muscle cells that surround the arterioles. And this is where we regulate blood pressure. So when blood pressure is low, these secrete renin, which activates the renin-angiotensin-aldosterone system. It also affects what we call the glomerular filtration rate, or how much filtrate we are forming. The higher the blood pressure, the higher the glomerular filtration rate will be. Okay, so as we see here, you have the proximal convoluted tubule, the nephron loop or loop of Henle, the distal convoluted tubule. Notice that it goes right up next to the afferent and efferent arteriole. So they have these specialized smooth muscle cells that surround them. Those are your juxtaglomerular cells. And those come in close contact with these straightened squish cells, the macula densa of the distal convoluted tubule. And when the amount of filtrate that's in the distal convoluted tubule is low, they're going to release renin. When the amount of blood coming into the afferent arteriole is low, they produce renin. The collecting system collects filtrate from the distal convoluted tubules. And the collecting duct can basically be divided into two parts. The portion of the collecting duct that is still in the cortex, cortical, and the portion that extends into the deeper part of the medulla. It is simple cuboidal epithelium with very few microvilli. So it resembles the distal convoluted tubule. Okay, so the collecting system, a lot of your distal convoluted tubules will dump again into the cortical portion of the collecting duct. Then it changes into the medullary portion of the collecting duct. And at that point, I should actually probably draw it more like that. You have several of these that dump into this larger duct, which is the papillary duct. And the papillary duct is within the papilla or apex of the medullary pyramid, which is this very bottom outer portion. And it is at this point that the fluid within the tubule is no longer called filtrate. It is now urine. You cannot reabsorb things or secrete. And urine will then be drained by the small little funnels, the minor calyces. So you can have, again, multiple minor calyces that will dump into a major calyx. And then that will dump into the renal pelvis, which of course forces everything to go into the ureter. Wider. So the kidneys can have two different types of nephrons that have different structures to them, or they differ in their structures, and they actually function in different ways, their primary goal. And we primarily name them based on their location. So cortical nephrons are found within the cortex. Juxtamedullary are found right at the border of the cortex and the medulla. Cortical nephrons make up the majority, about 80%. So these will begin, the renal corpuscle will be in the most outer portion of the cortex, and the nephron loop or loop of Henle are very short. If they go into the nephron, or the medulla, they do very little. The capillary beds that surround these, aiding in reabsorption and secretion, are what we call peritubular capillaries. So they allow the exchange of materials from the tubule into the interstitial space, which of course you have fluid there, back into the capillary bed. Juxtamedullary, less common, about 20%. Again, they're found right at the boundary. They have very long nephron loops or loops of Henle that go very deep into the medulla of our kidney. They do have peritubular capillaries that surround the proximal and distal loops, but they have the straight ladder-like capillaries that extend and go around. I should have done that a different color. That surround the loop of Henle, or the loop, and those are what we call vasa recta. Both of these types will drain back into the interlobular or cortical radiate veins. So the juxtamedullary, having this much longer loop with additional capillaries, allows us to control the volume of urine, and concentration. And we will talk more about that later. Okay, so this is just showing you, comparing and contrasting. So again, notice the cortical nephron, the loop is very short. It does extend a little bit into the medulla, where the juxtamedullary, the corpuscle, is kind of right there at the boundary, and the nephron loop is very extensive, and you have very long vasorecta. Again, you do have the vasorecta. You can have it around the cortical nephrons as well. It's just not as extensive. Nephrolithiasis is the formation of kidney stones, or what we call renal calculi. Calcium oxalate crystals accumulate and they form hard spherical structures. These form when concentrations of ions are going to be much higher than normal, to the point that, remember, ions can form salts, which are crystalline structures, to the point we say it's super saturated. And so they form these hard spherical structures which can block urine flow. So the crystals typically form either in the nephron loop or the distal tubule, or they could form in the collecting system, either the collecting duct or the papillary duct. When they're very small, we can pass these easily into the urine. They can sometimes stick to the epithelia of the tubules, a lot of times in collecting. And unfortunately, when they start to stick, they form what we call seed crystals. And these will stay in the system. More crystalline ions will continue to stick to it, and it will get larger. It could break off and lodge these crystalline structures into the minor and major calyces, into the pelvis, and into the ureter. So, of course, when that happens, urination becomes very painful, which is one of the most common symptoms is painful urination, blood in the urine, sweating, nausea, vomiting, which sweating, nausea, vomiting kind of always go together. And then you have severe pain in the lower back and the renal area. Sometimes it even feels like it might be within the large intestine just because of its proximity. We call it renal colic. Things that would increase your chances of developing nephrolithiasis would include, if your family history, genetics, has a greater incidence of this happening, people that do not have enough water if they're dehydrated, diets that are higher in fats, which means you have more bile salts, and these bile salts can accumulate, people that consume great amounts of animal protein, salts, and people that are obese. Also, people that have hyperparathyroidism, or if you have a renal infection, that could increase your risk as well. So how they will diagnose this, they will do a CT scan or an intravenous pyelogram to see if they can see the stone. And in this case, as we see this little stone right here, it's completely basically blocking the ureter. The size of something like that, they're more than likely probably going to have to surgically remove it. Sometimes they can use lasers to break up the kidney stones into smaller pieces that can be passed. Okay, review over some of the anatomy. Urine collection occurs within the pelvis of the kidney. So the cortex and medulla contain portions of the nephron, which is where urine is made, and the columns are between the medullary pyramids, which are in the medulla. Blood exits the glomerulus through the efferent arteriole. E for exit. Afferent brings blood to the glomerulus. Renal, we don't really just have a renal arteriole, you have a renal artery. Peritubular refer to the capillary beds that surround the nephron loop. And then arcuate, or what arch over the renal pyramid. From the arcuate, you have the interlobular or cortical radiate, and it's from those that you have the afferent that feeds into your glomerulus. Blood filtering structure of the kidney is called the glomerulus. That is the capillary bed, and really I would say renal corpuscle to be more clear. You can't just have the capillary bed, you have to have the surrounding capsule that can collect the filtrate. Urine forming units in the kidney are the nephrons. Even though technically the nephron doesn't make urine, it makes filtrate. Once that filtrate reaches the inferior portion of the collecting system, the papillary duct, it will become urine. So basically whatever's in the urine are harmful substances to the body or ions, things that we have in excess. The vasa recta are associated with the long nephron loops or loops of Henle, and those are what we have in the juxtamedullary nephrons that have the long loops. The juxtaglomerular apparatus is made up of JG cells or juxtaglomerular cells. Those are the specialized smooth muscle cells that surround the afferent and efferent arteriole and the macula densa cells of the distal convoluted tubule. Remember, their function is that they regulate blood pressure and glomerular filtration rate. Juxtaglomerular nephrons, we're going to talk more about this later, but the purpose of the nephron loop is that they allow for more water and salt to be absorbed, which would make your urine more concentrated, would help raise your blood volume. 80 to 85% of the nephrons in the kidney, that describes the cortical nephrons. Renal corpuscles in the outer portion of the cortex also describes cortical. So does the very short loops of Henle. Which of the following labels correctly identifies the juxtaglomerular apparatus? That's going to be D. So again, that includes the macula densa cells and the juxtaglomerular cells. A would represent the parietal layer of Bowman's or the glomerular capsule. E is your afferent arteriole. B is the efferent arteriole. And then C would be the distal tubule or distal convoluted tubule.